Okay, so when we're talking about neurons, or really any cell, when we're talking about the resting membrane potential, so where the cell is hanging out just during its normal function, our resting membrane potential for the neuron, for question 1, is going to be negative. It's important to keep this in mind because that resting membrane potential, the fact that it's negative, is what creates the electrical forces on our ions. So any positive ion, the electrical force for those positive ions is going to be into the cell because they're attracted to that negative charge. Because the inside of the cell is negative, that also means that any negatively charged ion, so any anion, will be repelled from the cell. So that those negative ions will want to move out if we're just thinking about the electrical force. Now remember that we have to track the electrochemical force and that our chemical gradients also make a difference when we see how the ions actually move. But this resting membrane potential always tells us about the electrical component of that force. So for our second question, we're talking about the resting membrane potential. We're thinking about the chemical driving force on potassium. So our chemical driving force on potassium, correct, is out. So we have a lot of potassium. We have a high concentration of potassium inside the cell. So our chemical driving force for potassium is towards that, from that area of high concentration inside the cell out to our area of low concentration outside the cell. And we've tracked how that difference in concentration is really due to our sodium-potassium pump. So we've been using ATP in order to create and maintain this chemical gradient. So for our second question, the resting membrane potential, chemical driving force on sodium, correct, is in. So this is the flip side, right? We've been setting up a sodium gradient and a potassium gradient. The sodium gradient we've been setting up with our sodium-potassium pump is that we have a lot of sodium outside the cell. Remember that we export 3 sodium when we run our sodium-potassium pump. Okay, so we have a high concentration of sodium outside the cell. We've been getting rid of all the sodium inside our cell. So that sodium wants to move down its concentration back into the cell when we're looking at the chemical driving force. Now we get into adding in our electrical forces, right? So we're going to refer back to question 1. Remember the resting membrane potential for our neuron is negative. So when we're thinking about the electrical forces, our first question, question 4, is the electrical force on sodium is blank the cell. Most people got this correct. So our electrical force on sodium, sodium is a cation, sodium is positively charged. Our resting membrane potential is negative. So our sodium, the electrical force, is into the cell towards that negative charge because our opposite charges attract. Now for the second part of this question, we're trying to figure out what this does to the movement of sodium. So really we're talking about how this changes the rate of that movement. So here for the second part, we're comparing basically whether that electrical force is competing with our chemical force or whether it's going in the same direction and therefore kind of magnifying that force. Now for sodium, if we look back, we saw that the chemical driving force for sodium is into the cell. And now we've seen that the electrical force on sodium is also into the cell. And that means there's no conflict when we're looking for our electrochemical gradient. They're both moving into the cell, right? We have a high-low gradient of sodium high outside the cell, low sodium inside the cell going in. We also have our opposite charges attracting, positive sodium outside the cell, trying to get to the negative charge inside the cell. So they're moving together into the cell. And the way we figure out the electrochemical driving force is we just add those charges together. And since we're going in the same direction, they basically add up to a number that's larger than either the chemical driving force or the electrical force on its own. Okay. So our final question, tracking the electrical force on potassium and what that's doing to the rate of the potassium movement, is a little trickier. So if we look back up, we saw that our chemical driving force on potassium is out of the cell. So potassium wants to get out because there's not very much potassium, or sorry, there's a lot of potassium inside the cell. There's not very much potassium outside the cell. But the electrical force is in the opposite. Oof. Oh, sorry, I see the problem. The orange one is highlighted. Okay, so the answer to this fifth question is actually C. So the electrical force on potassium is into the cell. Oh, sorry, D.
Are you?
Sorry, I flipped up my answers.
You intend to share your screen with us?
Oh, is it not sharing?
Yeah, no.
Can you see the poll results? Oh, okay. So there's no like correct answer written down for polls. So I'm just explaining the answers, and then we'll see on the slides how they match up with the figures. Sorry. I'm looking at my personal notes. Okay. So I was explaining correctly. I got mixed up by the orange poll result for potassium. Okay. So when we're thinking about potassium, we saw that for potassium, we have a lot of potassium inside the cell. We don't have very much outside the cell. So looking back at number 2, we know the chemical force is out. We know that potassium is positive and the resting membrane potential is negative though. So the electrical force on potassium is still into the cell, just like it was for sodium, because all we're thinking about when we're looking at electrical forces is the polarity of those charges. So whether they're positive or negative. So the electrical force on potassium is into the cell, because all we're thinking about here is positive and negative. We don't have to think about the concentrations at all. So it's into the cell, which is the opposite of our chemical driving force from question number 2, which means that because our chemical driving force and our electrical driving force are in opposite directions, this means that the electrical driving force is basically slowing down the movement of potassium. So because they're in opposite directions, if you added up the forces, basically you'd have one number that was a positive because it's going one way, but your other number would be a negative because it's going the opposite way. So when you come up with a number for the electrochemical driving force, it would be smaller than our original force. And now I am going to share my screen and we'll look at the pictures of the cells that explain all of these things. Okay. Okay. Oh, quick reminder, as I'm scrolling through those, you do have a mastering A&P assignment coming up. So we're doing mastering A&P for chapter 8. So write that down in your notes. If you haven't done it yet, make sure you get to those before the next class. Okay, so here are the pictures that explain what we're talking about in these polling questions. This has no membrane potential initially because this is showing us how we set up those chemical gradients. But our chemical driving forces, we can see from the relative concentrations here. So here in this picture, we see that we have high potassium inside the cell. In this picture, we don't have any potassium outside the cell. And we see the reverse for sodium. So we have high sodium outside the cell. And we don't have sodium inside the cell. So this is showing us on the top part, it's showing us the chemical force for potassium being out, the chemical force for sodium being in. Okay. We talked very briefly then, or we talked about this last time we met, about how the fact that the membrane is more permeable to potassium means that potassium basically wins out when we're talking about which of our ions moves more. More potassium moves out of the cell than sodium gets to move in. So the inside of the cell becomes negative and moves towards potassium's equilibrium point, which is -94. Okay, so that gets us to the setup where we have our resting membrane potential negative inside the cell. And so then we can start to think about these electrical forces. So here's this part where we see our sodium basically speeding up, the electrical force speeding up the direction of the chemical force, because we can see that our chemical force for sodium is just the same as it was on that previous slide. So that chemical force for sodium is into the cell. And our electrical force on sodium, our positively charged sodium, also into the cell. So we can see how they're lining up together like that. So they're going the same direction. So they're complementary, they add together. On this top half of the image, we see what's going on with the potassium. So we can see that our potassium chemical in yellow, right? The chemical force for potassium is out. The electrical force for potassium, because it's positively charged, is into the cell. So we can see that that's competing with this chemical force. Now the chemical force is stronger than the electrical force. That's why this yellow arrow is bigger. So the chemical force is going to win out. But this electrical force is basically going to slow down this outflow of potassium. So I want to pause here because it is clear that there's a little bit of confusion on this. Does anybody have a question that they can articulate about how these chemical and electrical forces are kind of working together? Because this is important to get down before we move forward.
I think what's throwing me off personally, the word right now, is trying to like keep separate the sodium potassium pump from like how this is working now. Okay. I keep switching the two.
Okay, I hear that. In that case, as we're moving forward talking about this resting membrane potential, the sodium potassium pump was important for setting this stuff up, but we can kind of forget a little bit about the sodium potassium pump for now if we want to do that, because all the stuff that happens later is happening after we set up those gradients. So if you want to just remember what the chemical gradient is without thinking about how it got that way, then maybe that'll help a little. All right, maybe practice just drawing these forces out, because this picture really just summarizes everything that's happening at our resting membrane potential, because we got our chemical forces on there and we got our electrical forces. Okay. So as we think about this resting membrane potential, we do have a specific number that we attach to this, which is -70. So this is just a fact about our neurons, and it's influenced by the number of channels we have for potassium versus sodium. So the reason we have this -70 specifically is because we're getting pretty close to our equilibrium potential for potassium, because our membrane is made more permeable to potassium by having a greater number of potassium channels. We do develop a steady state where at -70, we essentially have the same number of sodium ions coming in as potassium ions going out. And then we kind of chill out there. This is where we're in balance, that this -70 is pulling hard enough on the sodium into the cell and hard enough on the potassium into the cell that it's evened out those rates of the sodium flow to the potassium flow. Sorry, I think this is the last time we'll see the sodium potassium pump. But the fact here just is that the sodium potassium pump is going to like maintain our concentration gradient so that we can stay having enough of a chemical force in the appropriate direction to maintain this membrane potential. Okay. So it's just maintaining the status quo here once we're at -70. Okay. Another way we can think about the resting membrane potential for our neurons is by basically coming up with this little chart of the equilibrium potential of sodium versus the equilibrium potential of potassium. So you see here on the top, we have our equilibrium potential for sodium at +60. So we remember that at the equilibrium potential for sodium, this is where our chemical force and our electrical force would kind of balance out. Our sodium is always trying to make the membrane potential of our cell reach its equilibrium potential. So sodium is trying to move into the cell pretty continually because it's trying to get that cell to +60. It's pretty far away from our membrane potential. So it's pulling pretty hard towards positive 60, but it's being outcompeted by our potassium equilibrium potential. So our potassium equilibrium potential is down at the bottom here. So our potassium equilibrium potential is negative 94. So our potassium is pulling and pulling and pulling and pulling, trying to get that cell to negative 4. It doesn't win totally. So we can see our resting membrane potential, not in the middle in terms of half, but like on the lower half of this, somewhere in between sodium and potassium. And we can see that resting membrane potential at negative 70. It's pretty close to our equilibrium potential for potassium, but not all the way there, because we have both the pull from potassium and from sodium, and they're pulling in opposite directions. Okay. When we're thinking about the forces acting on ions, there's always going to be some kind of pull for that ion if the membrane potential is not at the equilibrium potential. So the fact, so flipping back to this diagram, the fact that we're not at positive 60 means that sodium is going to have some kind of a pull, some kind of a force. The fact that we're not at negative 94 means that there's always going to be some kind of force on our potassium as well. Okay, so our electrochemical force is not 0 for either of these ions at the resting membrane potential, even though we have a balance between the sodium and the potassium. So each ion independently always is going to want to move. And the net force acting on that ion is going to be in the direction that favors its being at equilibrium. So I'm going to flip again back to that little chart here. So we're always going to want to move towards our own equilibrium potential if we're an ion. So sodium is positive. Sodium always wants to make our resting membrane potential positive. So sodium is always going to want to move into the cell because it's positively charged. So it wants to get rid of this negative charge, basically. The opposite is true for our potassium. Potassium is positively charged, but it wants the inside of the cell to be even more negative because its equilibrium potential is even more negative. So in order to do that, potassium has to escape the cell. Okay, so that's what the second bullet point is saying, is that the net force on the ion is going to be in the direction that gets it to its equilibrium potential. This final factor we haven't talked about yet. So we have talked about the electrochemical force and the equilibrium potential before, but we haven't talked much about the strength of that force. And a fact that is true is that the strength of the force is going to be increasing the farther away from that membrane potential we get. Okay, so if we look back at this diagram, we can see we're really far away from the equilibrium potential for sodium. We're not that far from the equilibrium potential for potassium. So there's going to be a stronger force on sodium than there is on potassium because it's farther away from the current state of our cell. Okay. The next two slides are going to kind of calculate in numbers how that works. So our resting membrane potential is at negative 70 millivolts. And if we're looking at potassium, I'll look at potassium first. Our potassium equilibrium potential is negative 94. This means that we're a total of 24 millivolts away from the equilibrium potential for potassium. So we can see, right? We need to become 24 millivolts lower in order to reach that equilibrium potential. And this basically means that our force is pretty weak. So the equilibrium for electrical force is into the cell. Our chemical force is out of the cell. And our chemical force is stronger than our electrical force. And we practiced a couple of those problems in our last unit. So this is that thing where we were comparing our equilibrium potential to the membrane potential to figure out which of these forces is stronger. Okay. So our net force on potassium is pretty weak. Potassium flows out of the cell, but it's important to remember here that our membrane is highly permeable to potassium. So potassium still has a really strong effect on our membrane potential because the potassium actually can move. So we have a lot more potassium channels, which is part of why we have our resting membrane potential much closer to the potassium equilibrium potential than the sodium equilibrium potential. So even though this force isn't that strong, the equilibrium potential of potassium pulling on our resting membrane potential, it kind of wins out because the membrane is 25 times more permeable to potassium. So it like has the ability to actually do something. For our sodium, we have a much stronger force. So our resting potential is at negative 70 millivolts. Our sodium equilibrium potential is at positive 60 millivolts. So we're a whole 130 millivolts away from the equilibrium potential for sodium. So we need to get to 0, right? 70, plus then we'd need to go another 60 past 0 to get to our equilibrium potential. If we compare the equilibrium potential and the resting potential, we find that the electrical force is into the cell, right? This positive charge on sodium attracted to this negative charge. Our chemical force is also into the cell. These two factors together mean that our net force is really strong. So sodium really, really, really wants to get into the cell, but it can't. It can't because this membrane isn't very permeable to sodium. So even though we have this strong force for sodium to flow into the cell, we can't have that much of effect on the resting membrane potential because those channels are just full. They mostly don't exist. So even though we have this strong force, it can't be released. And this is really important to remember because as we start talking about action potentials, we will be talking about situations where we change the permeability of the membrane to sodium. And then we're going to see this really strong force released. Once we change the membrane and make it more permeable to sodium, we'll see that sodium can flow into the cell and it's going to rapidly get us up much closer to its own equilibrium potential. So at certain points in our action potential, we're going to see a ton of sodium flow into the cell because of this strong force. Okay. But at rest, we have very little sodium leaking because we have a high force for sodium, but the membrane isn't very permeable to it. We don't have very much potassium leaking out of the cell because even though our membrane is pretty permeable to potassium, we don't have very much force because we're almost at the equilibrium potential. And we remember that those chemical and electrical forces in potassium are competing. We have the sodium pump that's just maintaining our gradients, keeping those forces in this direction in this situation, making sure we have sodium and potassium in the right places. We're gonna, I think I can do this. Okay. I'm gonna switch my sharing options. We're gonna watch this short video of it being set up because I think seeing it in kind of 3 dimensions and watching them move might be a little helpful.
The human brain alone contains about 100 billion nerve
Sorry, I'm gonna pause this. Can you guys hear this playing? I just wanna make sure I'm sharing it right. Okay, great.
cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay. Super scintillating video, but that's what it looks like if we look at the movement of our sodium and our potassium moving through their respective channels in order to get to our resting membrane potential. So a good thing that video brought up also, you may not have keyed in on it, it was calling these channels leak channels. And that is true. We're going to see some other different types of channels. So maybe put that in your brains now. The leak channels are the ones that are basically always open, helping us maintain the resting membrane potential. We have a big slide comparing all the different types of channels coming up in a bit, but these ones that we've been talking about are the leak channels. Okay. So I think we're gonna let these facts about the membrane potential kind of percolate for a bit, and keep thinking about those, keep kind of practicing. I recommend doing some little drawings of how all these ions are moving to maintain the resting membrane potential, because understanding our setup for the cell and the forces acting on these ions when we're at rest is really going to help us understand what's happening as we start to change the membrane potential during our action potential. So this is a diagram of what our action potential is actually going to look like. So we're going to see that there are going to be a series of phases where we change the membrane potential of our neuron. Okay. So what we've been talking about is just setting up our resting potential. So we can see this dotted blue line at -70. So that's what we've been talking about, setting up with these chemical gradients and the electrical forces on our sodium and potassium. So this is the purple line where we don't have really anything highlighted. So moving forward, we're going to move into talking about our action potential. And we're going to see kind of this waveform developing. We're going to see in the next couple slides how we get up to a threshold. So we're going to start changing the membrane potential, and we're going to get to this threshold of -55 millivolts, and we'll talk about how that happens. But then we're going to have something called the action potential form. And basically what we're going to see happen is that we're going to allow ions to move such that we raise our membrane potential up positive, positive, positive. We get to +30. Then we're going to drag that membrane potential back down, back to -70. We're actually going to overshoot it a little bit before we return to the resting potential. So hopefully in our minds now, kind of knowing which direction sodium and potassium want to go, we can even start to think how this would work, right? We remember we have our sodium equilibrium potential way up here, right? It's even farther, even more positive than +30. We have our potassium equilibrium potential way down low. It would be at like -90, -94. So hopefully we can kind of predict that one of the things that we may have happen in order to make these changes happen is that we'll see that we're going to allow sodium to take control at one point, then potassium to take control, even more control than it usually has at another point. So we're basically going to be playing with the permeability of sodium and potassium in order to get these spikes of electrical activity. These spikes of electrical activity in our neurons is really how our neurons are able to signal. We're diving into that in detail next, but I just wanted us to take a quick look at that right now. As we're talking about our neurons and these signals, I also want to quick go over the anatomy of a neuron, because I know we talked about it a little bit in physiology, but not in very much detail. And this will all make a little more sense if we kind of know where we are in the neurons. Okay, so we talked about in physiology the fact that the anatomy of a neuron involves a cell body. So we have this central cell body. We see this sort of pink-purple nucleus. We're now going to call that the soma of the neuron. So the soma of the neuron, and soma actually always means body. It's just a word for body. Somatic cells are cells specifically of the normal parts of your body. So the soma is just the cell body of the neuron. We're also going to see that we have dendrites, these incoming signals. So you can see in green, there are some lines moving down these kind of hairs or tendrils coming off the soma. Those are our dendrites. This is where we're going to have signals coming in. They're going to deliver signals to the soma, to the cell body. And then we're going to have a whole apparatus that sends our signals out. So I want us to note this kind of swelling, basically the sort of triangle pyramid shape as we move into our long sort of strand of the axon. This is called our axon hillock. So this sort of section of the soma that starts narrowing off, moving into the axon. It's our axon hillock. So as we talk about the action potential, this is where we're going to be starting out. We're going to see our action potential start right here, right before the beginning of the axon. Then we'll talk about the axon itself, which is where we're going to send our spike of electricity out. We'll see how that spike kind of travels down the axon in a wave. And then as we start talking about synapses and neurotransmitters, we'll start talking about the axon terminals, which are these kind of noodly fingers at the end. That's where we're going to have the release of neurotransmitters to signal to whatever cell we're joining up with, whether that's like a muscle cell or another neuron. Yeah. I just want to get that in our mind because we've been drawing cells so far as little circles, and we will continue drawing them as little circles fairly regularly in order to talk about membrane potential. But as we move forward into signaling, it's going to become a little more important that we understand the shape of the neuron, because different things are going to be happening in different parts. Okay, we got a couple more minutes. We're going to flip into our action potential itself. We're going to start out by talking about graded potentials. And then we're going to move into how that leads us into the signal that we're going to send down the neuron and eventually manage to get our signal out to the rest of the body. Okay. So yeah, I'm actually going to put us into groups for a second, and we're going to talk about predicting what's happening during depolarization. So one thing that we're going to see happen is that when we get a signal from, for example, another neuron, we'll see that the different neurons can link up together in pathways. Sometimes what's going to happen is that a neurotransmitter is going to bind to a receptor. So a receptor is just a protein on the outside of the cell. And binding to this receptor is going to change the expression of channels in our membrane. And one of the things that can happen is that our neurotransmitter opens sodium channels. When that happens, something called depolarization happens. And we want to think about what change this is going to have on the membrane potential if we have a lot more sodium channels open. And we're also going to want to sort of compare that to what would happen if we have potassium channels open. I'm going to move you into a couple breakout groups, and I want you to talk about this for a second. You can use your whiteboards to draw. That might be helpful. And we're going to circle back. Okay, we just got a couple more people to come join us. They'll be here in the next few seconds. I'm going to start out by drawing us a nice little cell to show what's happening. Okay, looks like we got everybody with us. Okay, so what we're talking about, we have our normal cell. So it's going to start out at -60. Then we're going to have our neurotransmitter come in. We're going to think about this right now as a signal. So we're just going to think about it as this neurotransmitter kind of zapping our cell. So our neurotransmitter is coming in. What it's specifically doing is going to open a bunch of sodium channels. Okay, so who wants to tell me what's going to happen when we open a bunch of sodium channels?
I said it would correspond with like phase 1 and make the membrane super positive.
Exactly. And why is that? How is the sodium moving?
There's more channels making it go inside the cell because it's negative.
Yeah, exactly. The inside of the cell is negative, so we have that electrical force for sodium moving inside. We also have our chemical force for sodium moving inside, so we have a super strong force for sodium going into the cell. So we have sodium, which is positive, moving into the cell, which means our membrane potential will become more positive. So now next we're thinking about a cell where we have a neurotransmitter that instead it zaps us, right? This neurotransmitter doesn't open sodium channels. Instead, it's going to open potassium channels. Now we already had a bunch of potassium leak channels open, but it's going to open even more channels. So it's going to happen to our membrane potential now as we increase permeability to potassium.
It'll get negative because we have a lot of potassium in the cell and it'll start to leave.
Exactly. Our potassium is going to leave the cell, which means it's going to become more negative. Okay. We could also think about each of these with respect to our equilibrium potentials. So our equilibrium potential for sodium is positive. So when we allow sodium to flow, the cell becomes more positive. Our equilibrium for potassium is highly negative. So when we allow potassium to flow, it becomes highly negative. So we're just moving towards those equilibrium potentials. Okay. Closing this. I can see that we only actually have 1 more minute of class. So we're going to start out next time talking about our graded potentials leading into our action potentials. But I want you to keep in mind the action of these neurotransmitters on those membrane potentials, because that's how we're going to start out. When we start out with these graded potentials, we're going to be talking about changes in charge. And I want us to realize that those changes in charge are often going to be due to neurotransmitters coming in and having an effect on our channels.